Good. So at the end of last lecture, I began to introduce the science of systematics, and to the modern science of systematics we're going to get to today, the means by which we reconstruct the pattern of evolution, the history of life. And as I mentioned before, Darwin has this metaphor of the tree of life. This is actually his first drawing of one. It doesn't represent any real particular example. It's sort of like a hypothetical case in his notebook. And the idea of the tree is that a trunk of the tree represents the shared common ancestry, ancestors and descendants through time. That when you see branching, that's divergence of that ancestral lineage into two or more branches, and that these branches continue out to tips, which either represent extinctions, lineages that have gone extinct, or the current species of the world. And in The Origin of Species, so 1859, Charles Darwin, in chapter 13, proposes that a new system of classification should be developed to replace the Linnaean one, or at least to modify it, based specifically on the concepts of common ancestry, or as he said, because it was Victorian times, propinquity of descent, but common ancestry. And biologists immediately failed to get on that. So they didn't. There were some people who thought about it, but they didn't actually come up with a good working system in the 1850s or immediately thereafter. So this is sort of a hypothetical case too. We've got the first living thing, we've got the last common ancestor of two different branches, then splits apart, and each of those diverges, and some are extinct and some survive to today. Now, our name for one of these family trees is a phylogeny. Phylogeny. That term was actually coined by the scientist who did this graph here. And you don't need to know his name is Ernst Haeckel. He was one of the first German advocates of Darwin's natural selection. So here, this figure here, this monophyletic branching tree of organisms, published in 1866. So the origin was only 1859. Quite frankly, it was at the end of 1859. So he really got on that right away. So a phylogeny, or family tree. Now, in principle, we would like to have a family tree, a phylogeny, that actually has direct ancestors and descendants on it, and tie those ancestors in with their descendants. But the problem that people began to recognize, particularly in the 20th century, was that IDing ancestors is difficult. Because remember, an ancestor is not merely an early relative. Your great-uncle is not your ancestor, but your great-grandma is. An ancestor is someone for whom you are an actual direct descendant. But the difficulty comes, for instance, with fossils. When you find a fossil, is that a member of the population that was directly ancestral to some later form, or is it just an early relative? So here we have the two closest living relatives at the top here. Homo sapiens, the only surviving member of the genus Homo, and the two living species in the genus Pan, so bonobos and chimps. And let's take the example here of Paranthropus. So Paranthropus, a so-called ape-man, so a creature more closely related to us, based on a number of attributes, than it is to chimps or bonobos. It existed earlier than us, and some people might be tempted to say, okay, it's our ancestor. But it co-occurs at the same moment in time, some of the same horizons, as another form, Australopithecus, which itself has even more traits that are shared with us, that are shared with Paranthropus or with Pan. And so one might say, okay, so Paranthropus is a side branch, but therefore, because it has more traits shared with us, then Australopithecus has to be our ancestor. At which point, you have to say, did you just frickin' miss the point? The problems we had with Paranthropus still exist with Australopithecus. We don't know that it actually is the population, or a representative of the population, from which we are actually directly descended. That is extraordinarily difficult to demonstrate, particularly when we lack genomic data, which is indeed the vast majority of the fossil record. However, So it might be this case. It might be that Australopithecus also is a side branch, and our ancestor is yet a third creature that was living at that time. So how do we demonstrate that? We know the fossil record is sparse. We know that the vast majority of species do not wind up in the fossil record. So how do we deal with that? Well, we stop obsessing with direct ancestry. And we start thinking about the pattern of evolution, and specifically, patterns of recency of shared common ancestry. That's something we can be more secure about. So we could be more secure based on the data that Australopithecus and Homo are more closely related to each other, that is, share a more recent common ancestor with each other, than either does with Paranthropus. That ancestor might have been a member of the genus Australopithecus, but it might not. But we could be more secure about this, and that Australopithecus and Homo and Paranthropus collectively share a more recent common ancestor with each other than any of them do with Pan. Whether any of these are direct ancestors of each other or not. So we can be more secure about this. And this was the intellectual shift in systematics, in the understanding of determining, or at least assessing, evolutionary relationships that occurred essentially a century and a decade after Darwin said that someone should do this. So it was developed in the mid-20th century by this guy, Willy Hennig. It's called cladistics or phylogenetic systematics. So cladistics comes from the word clade, which is already being used as branch. Technically, it's from a Greek word klados, meaning a stem or twig or branch. As a method, it was developed initially by Willy Hennig here, who was a mid-20th century East German entomologist, so someone who works on insects. Specifically, he was a dipterist. He worked on flies. He referred to it as, and we still call it, phylogenetic systematics. So a method of systematics that focuses on family tree relationships. And it's still the current primary method of reconstructing evolutionary relationships. It's been modified greatly ever since. We've been many advances, both in the types of data we use and the mathematics we use to assess these relationships, but it's still the dominant method today. Whether it's folks in this building who are using DNA and they've never seen the organism that they're actually encoding in there, or people like me who are morphologists who actually look at the organisms and measure them, even though we have less data because we don't use the DNA, or what have you. So, cladistics. Recognizing that direct ancestor-descendant relationships are difficult to test. Hello. It's hanging up here. What's going on? Let's see. Instead, again, relies on patterns of recency of common ancestry. And the branching order that we find on these diagrams is inferred by shared evolutionary specializations. So they are transformations from the old state. That's what makes them specialization. And they're shared ones. Or, as we will call them in more detail, shared derived characters. By the way, this word character, that is just a synonym for trait or attribute. Characteristic is actually an adjective form that somehow got transformed into a noun, but it's not. The proper noun is character. And this sort of thing is in drama, a character. You would take on, literally, a mask. You would take on the trait in ancient drama. You would wear masks. They would wear masks while performing. You would take on the trait of the persona you were presenting. And so that became our dramatic use of the word character. Derived means it's transformed from the ancestral version. And shared, again, because it's only those traits which are shared among multiple creatures that are useful for determining evolutionary relationships. So on this diagram, no biologist in the world would disagree with the relationships here. I'd be very, very hard-pressed to find one. It does not say that dinosaurs, a Tyrannosaurus rex, is a descendant of a modern shrimp. or that modern shrimp and dinosaurs are the descendants of cacti, or that all those animals and plants are the descendants of bacteria. But it is saying that shrimp and dinosaurs share a more recent common ancestor with each other than either does with plants, and that plants and animals collectively share a more recent common ancestor with each other than any of them do with bacteria. We'll see how that works in more detail. So patterns of common ancestry. Any two organisms will have some most recent shared common ancestor. Now, of course, they will have even earlier shared common ancestors. All living things have a shared common ancestor with the very first living thing to evolve. But that's not helpful. But any two organisms will have some most recent, that's closest to us in time, shared common ancestor. But that statement in and of itself isn't super useful. Because it's true that lions and tigers will have some most recent common ancestor, but it's also true that a lion and you have some most recent common ancestor, and that a tiger and a rutabaga has some most recent common ancestor. So it is a trivial statement to point out that any two organisms have. But when we have multiple organisms to deal with, then we can start to get a pattern. So for instance, we could recognize that all the creatures here share a trait of the presence of a bony skeleton. Most organisms don't have a bony skeleton. Most animals don't have a bony skeleton. And many vertebrates don't. Sharks don't, lampreys don't, hagfish don't. A subset of these groups don't merely have a bony skeleton, but they also have limbs that have fingers and toes. And a subset of these also have fur and give milk. And then a subset of these have retractable claws as well. And even at the time of Darwin, he was beginning to notice that what we're seeing here likely reflects the evolution of new traits that were then passed on to subsets of these larger groups. Now, I could have drawn these as a Venn diagram, a series of nested sets. And for small collections of taxa, of organisms, that might not be too bad. But mathematics gives us another way of displaying the same information, and that's a branching tree. So the branching tree is just a representation of the sets involved. It's just easier to draw, particularly when we have lots and lots of forms. It also has the nice aspect of reflecting this idea of a tree of life. And as we'll see at the end of this lecture, which hopefully will happen in this, this lecture block, if I don't quite get to it, I will record it on Canopto so that we can start fresh on Friday with the second section of the course. And this sort of stick figure diagram is called a cladogram, a branching diagram, a cladogram. And it represents a stick figure simplification of the actual family tree. So it's saying, well, we'll see in a bit. So cladograms. And we're going to be using cladograms a lot in this course. So cladogram, a stick figure simplification of the actual family tree. And clearly, I've left out a lot of organisms. It is not the complete family tree. Now, a cladogram isn't quite the same thing as a true phylogeny. For people who make a distinction here, a true phylogeny is the actual tree that includes actual ancestors in there going earlier back. So you can see that lions and tigers are there as one group, and then lions, tigers, and bears. Thank you. And then lions, tigers, and bears, and cattle, and lions, and tigers, and bears, and cattle, and kangaroos, and then all those guys, and a duckbill platypus going further and further back. But I think you can see how we could simplify just the taxa we show on the top and the relationships in this branching diagram, this cladogram. Now I want to introduce a term here. I like to use this term. It didn't take off the way people hoped, but I like to promote it. And it's the word concestor. The concept is well known in phylogenetic systematics. It's just people refer to it as different things. I like the word concestor. It's a shorthand for most recent common ancestor. Some people will say MRCA. That's useful. I like concestor, it's a better word. And that's a specific set of ancestors. So there is the concestor of lions and tigers. Presumably, it would be a cat. In fact, it would be a member of the genus Panthera. It would not be an individual. It would be a population, because organisms descend from populations. We may never find that concestor, but we infer its existence. Earlier in time would be the concestor of lions, tigers, and bears. This would be the concestor of all carnivorous mammals, a group called carnivora, the carnivorous mammals. And even earlier would be the concestor of the carnivorous mammals and forms like the hoofed mammals. And earlier than that, the concestor of all mammals, and so forth. Okay. So, again, the trivial statement that any two taxa, so let's do some dinosaurs in this dinosaur class, that any two taxa have a concestor, even if we haven't found it yet. So, Gorgosaurus and Tyrannosaurus would have had some concestor, unless one of these were miraculously, created independently of all other life. That's a trivial statement. But if we add a third taxon, then we actually have a hypothesis. So this diagram is actually a hypothesis of evolutionary relationships. And according to this hypothesis, this little dinosaur, these aren't shown to scale. DeLong is about 1.5 meters, Gorgosaurus about 9 meters, Tyrannosaurus about 12 to 13 meters. So DeLong and Gorgosaurus and Tyrannosaurus collectively have a concestor. From that concestor, one lineage went to DeLong. The other lineage went to the shared concestor of Gorgosaurus and Tyrannosaurus, which then itself diverged into the lineage that contains Gorgosaurus and the lineage that contains Tyrannosaurus. Does everyone see how that works? Good. If you don't, as we're going through this, better to get to know how it works early, because if you don't learn how to read a cladogram, you're going to have a bad time. They're really going to be important in this course. Okay, so reading a cladogram. That's the cladogram I showed you before. This one, it's been flipped on its side. By the way, I should point out, most times we draw a cladogram, we have a little bit sticking off of it with no name on it, and that's just telling it that's towards the base of the tree of life. That's towards the origin of all life. It doesn't have a label on it. So this other one got knocked over on its side, but the information is there. And what does it say? Well, this one has the group of G and T. This one also has the group of G and T. That one has the group of D plus G and T. That one also has the group of D plus G and T. And I've now highlighted the G and T group with yellow. These are the same cladogram. One might be knocked over on its side, and if you note, the Tyrannosaurus here, you might think, hey, the Tyrannosaurus is between Gorgosaurus and DeLong, but it isn't. It's the branching pattern that's important. So it's this thing, D plus G plus T, and these both say the same thing. It doesn't matter that we spun it around here. That's not the information. Yes?
So the more recent one is going to be an analogy symbol, and that's the way you read it?
Not necessarily. They could all be co-occurring in time. Because remember the example of that I had with the lions, tigers, bears. Those all exist at the same time, and yet there's a real cladogram connecting them. Or how about this one? We see here a grouping of Gorgosaurus plus DeLong independent of Tyrannosaurus. So we try to reconstruct that G and T group. It doesn't exist on this other cladogram. DeLong stuck in between. So these are not the same cladogram. The groupings are the important thing. The branchings are the important thing. And this has a different branching pattern. This is saying Gorgosaurus and DeLong share a concestor that Tyrannosaurus didn't. Or you can think about it this way, that Tyrannosaurus branched off first. So here's a way to think about it. You've got the the part going to the base of the tree of life, working from the base, the order that they branch tells us the order of the branching events of that lineage. How about this one? Is this equivalent to the upper one, or is it equivalent to this one? Take a look. Yes?
It's its own unique cladogram.
Yes, I tried to trick you with that, and you got me. It is a third, mutually exclusive representation, because it's saying Tyrannosaurus and D. long had a more recent shared common ancestor with each other than either did with Gorgosaurus, which contradicts this and this. This is different from the other two. In fact, these are the three potential mutually exclusive, what we call dichotomous, so that's two-way splitting, cladograms that are possible. There is no other option possible, assuming dichotomous splitting. Now, if only we had some sort of way or a name for the idea of evaluating mutually exclusive hypotheses that concern the natural world. Oh, you know what's called science? That's the big breakthrough with cladistics. It changed systematics from sort of an opinion to science, to a method of hypothesis testing, by explicitly stating what the hypotheses you were going to test were. It also provided a methodology to do those tests, and that's what we're going to get to next. So, three mutually exclusive alternative tree topologies. So, for mathematics, tree shapes. How do we choose between them? Well, we're going to be looking at the presence of shared modifications. Now, for someone, as I said, in this building, they're going to look at patterns of shared DNA sequences for the most part, because there's just so much more data available. And you can actually let, throw the organisms in a blender or throw them in a machine. It will separate all those things out for you automatically. For those like me who work on morphologies, on shapes, we actually have to sit there, get out our calipers, make our measurements, describe them as accurately as possible to identify the differences. And of course, in my particular part of the field, we don't just include living animals, we include the fossil record as well. But the logic behind it is the same. And it's what we call phylogenetic analysis. So a phylogenetic analysis includes two sets of data. It includes the taxa, so the groups whose relationships are being evaluated, and the characters, or traits. So here we have those three dinosaurs we're looking at, but we need something else. We need something to help evaluate what the ancestral or earlier state is of each trait, and what the modified versions. Remember, it's shared evolutionary specializations. So what we need is something that's not in the in-group, that is the group studying, something that's an out-group. Some form that previous work shows, or at least suggests, is a relative of, but not a member of, the in-group. No, we could potentially be wrong, and it could wind up being part of the in-group. But you got to start somewhere in the study. And then we have traits that differ among the forms that we're interested in. So for each of these traits, the shape of the premaxillary teeth, the shape of the lateral teeth, so the maxillary and dentary teeth, the number of digits on the hands, the number of fingers, and the shape of metatarsal 3, the middle metatarsal. You don't need to know what these features are. You will eventually when we talk about Tyrannosaurus, but as the specifics here. But this is just an example, a super simplified example. And for each of these, the character, the trait, comes in 2 or potentially more states. To keep it simple, these all have 2 states, or binary states. In blue are what we call the ancestral or primitive condition. Primitive is not a judgment call in terms of its value in the universe, or anything. It's closer to the original condition. That's what the word primitive ultimately means, closer to the primal condition, or ancestral condition. So when we look at the wide variety of carnivorous dinosaurs, they tend to have cone-shaped premaxillary teeth, blade-like lateral teeth, 3 fingers on the hand, and an unpinched third metatarsal. That's true of Allosaurus, it would be true of Velociraptor, it would be true of many other forms. But we also try to find the derived, or specialized, or advanced condition. And again, advanced doesn't mean necessarily better, it means transformed from the ancestral state. In this case, D-long, Gorgosaurus, and Tyrannosaurus have premaxillary shaped teeth that have a capital D, or maybe a capital U is even better, cross-section. Gorgosaurus and Tyrannosaurus have thickened lateral teeth. They only have 2 fingers. They've lost digit 3. And they have a pinched third metatarsal. Okay. So, we take a tree, one of the 3 alternative hypotheses. By the way, those same 3 alternative hypotheses will be there. We've introduced Allosaurus, but remember, it's the outgroup. So, for the purpose of this analysis, it will always branch off before these other 3. And then we map the traits on there. So Allosaurus is our outgroup, and we are stating, for the purpose of the analysis, that whatever trait it has is ancestral. So those are the traits we would be expecting in down to this part of the tree. And then we look at the presence of the derived condition. So these 3 forms, remember the in-group, all have the derived trait. The derived state of the trait. Now, it could be that D-long evolved a D-shaped premaxillary tooth totally independently of Gorgosaurus and Tyrannosaurus. Or that D-long and Gorgosaurus and Tyrannosaurus all convergently evolved that trait separately from each other. Or that it evolved here and then it was lost in D-long's lineage and it re-evolved it, and then it was lost in the lineage going to Gorgosaurus and Tyrannosaurus and was regained in Gorgosaurus and was gained and lost and gained and lost and gained. Those are all theoretically possible. But there is a simple, easy solution, given this tree topology, to infer the origin of this trait. It is, in fact, the most parsimonious solution. Remember back to the nature of science, about parsimony, the principle of parsimony. All of the things being equal, assume the simplest solution for the observations you have. The simplest solution is that the trait for D-shaped premaxillary teeth evolved before or after the branching of Allosaurus and these guys, but before the divergence at the concestor of D-long, Gorgosaurus, and Tyrannosaurus, and was then passed on to all of its descendants. Did that make sense? Good. Okay. We can look at the next set of traits. Since D-long lacks thickened teeth, but both Gorgosaurus and Tyrannosaurus share it, the simplest solution is that that trait evolved after the lineage going to D-long split off from the lineage going to Gorgosaurus and Tyrannosaurus, and then was passed on to all of its descendants. And it turns out these other 2 traits have precisely the same distribution. We wouldn't know necessarily the order that these 3 came in, and it doesn't matter for the analysis. You know, it might have been 2-fingered hand showed up first, it might have been pinched metatarsal 3 showed up first, we don't know. They all occur on this branch, given this tree. And now, we can actually quantify this. Remember, that's another aspect of science, quantification. We can count the number of evolutionary transitions from primitive to advanced, from primitive to derived. And that's 4. Given these observations and this tree topology, the simplest solution is we require 4 evolutionary changes to explain this tree. Yeah?
I have a question.
Sure, sure.
Is it possible that, like, let's say here, that the long and Tyrannosaurus would share a trait, but Gorgosaurus wouldn't? What would you do with that?
Next slide.
Okay.
Or we'll give an example of that. So yeah, what happens if they don't fall out neatly like this? We'll see. So that was the tree I just showed there. But let's say it's one of these other trees. where Gorgosaurus and Tyrannosaurus had a bunch of traits in common that were not found in D-long, and yet we still observe them. So given this tree topology, the similarities between Gorgosaurus and Tyrannosaurus can either be explained as independently originating, that is convergently evolving in Gorgosaurus and Tyrannosaurus, or potentially that they showed up in the common ancestor of all these guys and then were lost or reversed to the ancestral condition in D-long. Anytime you have a convergent evolution of a trait or a reversal, that also counts as an evolutionary change. So we count that up. So in order to explain the same observations of what traits were present in these two trees, those require 7 changes. And that's true even if for each individual trait we decide if it's convergent or reversed, the number is going to be 7. So we have 3 mutually exclusive hypotheses with different tree topologies. One tree explains the data with fewer changes than the others. That's our preferred tree, or our most parsimonious tree. It requires the fewest numbers of assumptions. Is it the true tree? We don't know. You don't know these things. We infer these things. That's why we do analyses. We don't know the relationships. We infer them based on our observations. It is the best tree currently, given this information. But new information could require us to change it. Those could be new. We introduce new taxa in there, or we introduce new characters, and we rerun the phylogenetic analysis, and now a new set of relationships comes out stronger. Now, it's a 100-level class. This is the introduction of the concept. This is, of course, incredibly simplified. Normally, we have dozens of taxa and hundreds of characters that are evaluated. Humans cannot do this by ourselves. We use computer algorithms to sort this out. But the process is essentially the same. It's just the computer's doing it. Okay. Now we're going to move on and look at some other aspects of phylogenetics to expand this. And one thing I want to introduce now is that cladogram shapes can be different. And I don't mean the topology in the sense of branching events. I mean the way people draw them. This is the oldest and sort of traditional way of drawing them. And I will tell you, if I'm putting together a PowerPoint or on an Illustrator slide, it's a lot easier because each line segment requires 2 clicks and that's it. But folks from departments of education, departments of science education, actually study this for a living. They study the way information is conveyed. And they actually show that these sort of goalpost versions are easier for people to understand. Or rather, it's easier for them to not misinterpret. They recognize on these, when you have the goalpost versions, that we're looking mostly at divergences. So often, I will try to use this slightly more complex version that requires me to do a little more work. But sometimes I'll have these versions. I don't typically do these curly ones because those are hard to draw. And why the hell would people do this? Okay, on a diagram like this, it's kind of silly because there are so few taxa involved that why would you do this? But let's say you have dozens of taxa and you're trying to publish them as a diagram. It actually is a space-saving mechanism. It's the same information. I, as a paleontologist, am not as happy with it because in this case, time winds up being like concentric rings rather than the good old-fashioned stratigraphic order with oldest on the bottom and youngest on the top. But it's still technically the same information because we still have that little strip with no name on it that's going to the base of the tree of life. Okay, so let's see how thinking in a tree-based fashion can change the way we view and actually give us more information about evolution. Tree-based thinking is so-called because we stop thinking about kinds of organisms or types of organisms as if they were fixed things. And instead, start thinking about what the real pattern of evolution is, which is lineages of organisms, ancestors and descendants, diverging from common ancestors and evolving their own particular traits, step by step. Kind-based or typological thinking tended to get people stuck in bad mental habits, artificially grouping things together in their mind that actually were not more evolutionarily similar to each other, and making false conclusions about the evolution of, say, behavioral traits or physiological traits. I'll see examples of these later in the course. So, the first aspect of tree-based thinking is just this, the basic one. A cladogram is a stick-figure version of an hypothesis of a pattern of shared common ancestry. So I've already had a slide that stated that basically it's a stick-figure version of a hypothesis of a pattern of shared common ancestry. There we have the same statement again. So this diagram does not say that Gorgosaurus is an ancestor of Tyrannosaurus. It does not say that DeLong is the ancestor of Gorgosaurus and Tyrannosaurus. That's not what the cladograms say. What it tells us is that Gorgosaurus and Tyrannosaurus have a concestor not shared with DeLong, but that all three of them have a concestor together. And because of this, because Gorgosaurus and Tyrannosaurus have a concestor not shared with DeLong, this had to have occurred later in Earth history, because it itself is a descendant of that concestor. How much later? We don't know. But later. I'll show you the last couple slides for this set, which again might have to be reported. We'll show you one technique to use to assess when later it might be. But right now, I'll go relatively later. So this is not your family tree. You to your father, to your grandfather, to your great-grandfather, but rather going back to parents and grandparents and great-grandparents and all the descendants from that. And the same basic pattern with regard to evolution. Consequently, there's no such thing as an evolutionary scale. That is an old, outdated, typological concept that predates actually evolution. It goes back to pre-evolutionary thoughts about life and the status of beings in God's cosmos, and has no place in science. Now, on a cladogram, the branching pattern is the only important data, because that's what the cladograms are. So let's take the first set, the set on the left. These are the same cladogram, the same tree. Tree is a synonym for cladogram. So let's see. They both have a grouping of humans and cats. They both have a grouping of lizards and geese. They both have a grouping of humans plus cats plus lizards plus geese. And then they all have a grouping of those things with frogs, and then those things with a bony fish. What is that? They're trout. And we have the world's most cladogram-oriented version of Twister. This reminds you of that with the circles. So, does everyone see how that worked? Same content, just displayed slightly differently. So these are the same cladogram. The set on the right, your right, are not the same cladogram per se. Each one contains information the other one doesn't, but they don't contradict each other. For instance, they both have a grouping of humans and frogs, but A has the additional information that geese are part of that grouping and closer to humans than to frogs. They both have a grouping of goldfish and trout, but the lower one has some information of another fish, who I don't recognize exactly what it is, that's part of that grouping. And then they both agree that the bony fish and the land vertebrates form a clade exclusive of the grouping with sharks. Everyone see how that worked? Cool. So, yeah, you need to find the difference in pictures. They're the same picture. And for once, she's right. For once, she got it right. They are the same picture. So clades are nested within clades, which are nested within even larger clades, and so on and so forth. And these more, as we say, inclusive, ones that include more taxa, are pointing towards ancestors that are earlier and earlier and earlier and earlier and earlier back in Earth history, depending on the scope of what we're looking at. So we're looking at dinosaur evolution. Those will be points of ancestors within the Mesozoic. But if we were looking at the diversity of animal life, that would take us back, maybe before the beginning of the Cambrian. If we're looking at all life, it would take us back well back into the Precambrian. So cladograms show the branching order of organisms. They do not show the terminal taxa evolving into each other, nor do they show any sort of relative superiority. So I've already said all these things, just highlighting them so you keep it in mind. Also, outgroups are not ancestors. They are stand-in for ancestors to assess the transformations. This cladogram is not incorrect. But no one would argue that humans, or indeed all vertebrates, are ancestral to the echinoderms, the spiny-skinned organisms like starfish and crinoids and brittle stars and sea urchins and sea cucumbers. But vertebrates are close relatives of the echinoderms, and we could use the information from us to figure out what was ancestral to echinoderms versus what was the transformation within echinoderms. Now, the pattern of characters, the pattern of traits, is a record of their evolutionary history. So when we look at a tree, we see the distribution of traits, and that tells us something about the evolutionary history of the trait, and as we saw, we use that information to reconstruct the evolutionary history of those groups, or groupings. So because of descent with modification, characters will be passed on to descendants. But it turns out not all characters, nor all character states, are equally helpful in reconstructing evolutionary relationships. So, don't worry about writing, don't write this all down here. This is an equivalent to this is in the lecture notes. But just take a look at it. What do we see here? We see characters, and what we're seeing is the transformations. The red lines with a little name after it, word after it, shows the transformations, where they show up on this family tree, on this phylogeny. So let's just take an example of one. Amniotic tissue. A tissue that we'll actually address in Friday's lecture. I should point out, this lecture, part of it which will be recorded, I can look based on the time, is the last methods lecture. And starting Friday, we start tracking the history of life on the way up to the origin of dinosaurs. And we'll talk about this feature, amniotic tissue. A tissue inside the egg, or in our case, inside the body, that's used to allow the embryo to develop on land rather than the pond. Frogs and other amphibians don't have that. Fish don't have that. Non-vertebrates don't have that. But that trait evolved after the split between list amphibians and the land vertebrates, so-called amniotes, and was passed on to its descendants. Upright limbs, so limbs directly underneath the body, were present in the common ancestor of Eoraptor and birds, and was passed on to all of their descendants. So that's what those little tick marks mean. Now, when we're talking about primitive and derived characters, that depends on the scope we're looking at. If we're looking at all these land vertebrates, amniotic tissue is a derived trait, and in fact, it's a shared derived trait that unites the group we call amniota. Duh, that's where we get its name. But if we were just sorting out relationships amongst this group called Sauria, the presence of amniotic tissue isn't helpful. It's a primitive trait. It was already there in the first Saurian, the ancestor of all Sauria, and it's found in all of them. So it's not helpful there in sorting them out. Also, there might be unique traits. There often are. For instance, feathers are, so far as we know, only found in aves among the forms here. I'll say pennaceous feathers, and that would be a good, defensible statement. It's necessary to, we need to understand feathers to understand the biology of birds, but it doesn't help us sort out evolutionary relationships. If they and only they have it, then it's not helpful. I just said that. And now, getting towards something that spins off of what you were asking before in terms of sorting out evolutionary relationships, convergent characters are real. They do show up. Upright limbs are not unique to the Eoraptor plus aves group. They're also found in mammals, or at least in a subdivision of mammals. But the weight of the evidence shows that that evolved convergently. Similarly, a toothless beak shows up in the modern type birds. It evolved independently in turtles. And in fact, we have early turtles and early birds that have teeth. Now, convergent characters, I mentioned before, in terms of interpreting the biology of the organisms and their behavior could be useful. But I said that they might also be a problem, and now they're going to be a problem. The existence of convergent characters are problematic in phylogenetic analyses because they are positively misleading. And it's only by the weight of evidence that we recognize that they are convergent. Because, to be fair, when you're first seeing it, you don't decide that they're convergent. You recognize toothless beak, and you code them into your analysis as if they're equivalent. And then it's the weight of the evidence that shows that it evolved independently in turtles and birds. And then a special type of convergent character are reversals. So if you look at these modern vertebrates here, we see that hagfish don't have any sort of limb, neither fin nor limb with digits. And we see that the land vertebrates have limbs with digits, except for snakes. Snakes don't have limbs, and consequently they don't have fingers and toes. Now, in the case of the hagfish, limblessness is a primitive trait. It inherited limblessness from all its ancestors who never had limbs. All the ancestors down that way were wormy sort of creatures, basically. So, we don't expect it to have limbs. But snakes had limbed ancestors. So for them, limblessness is derived. We call that a reversal. A reversal is convergence on the ancestral condition. So just to summarize these again, shared derived characters. We like them. They're great. They help us tease out evolutionary relationships. But we don't know in advance if the trait we're seeing is a shared derived character. Shared primitive characters, not helpful in figuring out relationships. They're just the evolutionary leftover. So if we were trying to figure out if turtles are closer to lizards or crocodiles, the fact that they have a bony skeleton would be useless, because that's a primitive trait at that level. Unique characters, again, not helpful in sorting out evolutionary relationships, but they might inform us about the biology of the organism. Convergent characters might mislead us into thinking the convergent taxa are closely related, and similarly reversals might mislead us into thinking the taxa are closely related, and in fact it might, as you say, pull those taxa further down the tree, earlier branching than they actually were. And that's why we need to look at a diversity of traits and a bunch of taxa to be more secure in our results. Okay, so taking a look at this cladogram, which taxon has a rostral bone, a frill, post-orbital horns, but lacks nasal horns? Anyone want to take a stab? Yeah, go for it.
I would say Zuniceratops.
Zuniceratops. Does anyone have an alternative hypothesis? Okay, let's just double check. So as you march this up this tree, rostral bone shared by all these guys. Stegosaurus lacks a frill, then all these guys inherit the frill. All these guys get postorbital horns, but the nasal horn happens after the split between Zuni and these two, so in fact, you are indeed correct. So, I will end here. I will record the rest of this lecture so that we're not delayed, and that we pick up on Friday with the colonization of land and begin to march our way up the history of life towards the origin of dinosaurs, which will happen next Wednesday.